test? You want to test that volume there, Pete?
Sure.
Okay, good. Good. I think we're ready to go. 7:30, right on the dot. So Lori Schrader from the Yukon Science Institute asked me to introduce Pete here, and just to say a few words about the Yukon Science Institute and what it is. And the YSI is a non-profit organization that seeks to promote the public awareness of science in science activities in the Yukon and to aid in and facilitate scientific research and development. As a part of our mandate, YSA coordinates the lecture series that this lecture is part of. YSA is funded by the Department of Tourism and Culture, Heritage, and supported by the Yukon Beringia Interpretive Center. The Yukon Department of Environment provides funding that allows us to send speakers to Dawson and other Yukon communities. Just as a bit of advance warning, the next lecture will be Sunday, August 7th, and it'll be here at the Beringia Center, and Aisling Farrell, who is the collections manager at the La Brea Tar Pits in Los Angeles, California, will be here presenting on sort of the paleontology of the La Brea Tar Pits. But tonight's lecture, it's a real pleasure to introduce Peter Heintzman. He's a friend and a colleague from the University of California, Santa Cruz in the Paleogenomics Lab. And Pete is one of these whiz kids, and I'll call him a kid because he's younger than me, that uses ancient DNA preserved and permafrost preserved and other ancient plants and soils and bones, and using those to look at past environmental change and evolution and systematics of Ice Age beasts and other types of animals and plants. And Pete has a really cool, diverse background. So in our work with the paleontology program, we often engage with researchers that have, they've studied and published papers about woolly mammoths, but they've never seen a woolly mammoth bone or even care to, which is, I think, kind of a shame. Or
yeah.
They couldn't draw a woolly mammoth if their life depended on it. But Pete has a great background in biology and in paleontology that really kind of separates him from the crowd in terms of his diverse knowledge of organismal biology and also paleontology. And so he did his undergraduate degree at the University of Sheffield. He has his master's in paleobiology at Bristol, and his PhD at the University of London Royal Holloway. And Royal Holloway is quite a prestigious university in terms of their Quaternary, their Ice Age science program. So he's got really good pedigree, and that's why we're so excited that Pete gets a chance to work with us on a lot of our Ice Age material from the Yukon. And I should also say and give a big thanks and a warm welcome to Dr. Beth Shapiro, who's sitting right here. She can put her hand up. She is probably the world's leader in ancient genetic research, and she's brought, I think, 7 of her students from University of California, Santa Cruz on a wild western field trip through the Yukon. So they all arrived on a plane last night and are eventually making their way to Inuvik up the Dempster Highway with bypassing, going through Kluane country and going to Keno and going to Dawson. So she's taking these kids out of the lab and away from their computers for a while. I have gray hair. I'm allowed to call them kids, I guess. And you're a kid too. So so she's bringing them out of the lab and into the field and to see where some of this amazing stuff from the Yukon that often they get a chance to work on. So I'll leave it at that and introduce Dr. Peter Heintzman for our lecture tonight.
Well, thanks, Grant, for that very comprehensive introduction. So today I'm going to be talking to you about ancient DNA that we have extracted, sequenced, and used to infer past biological processes from 2 different taxa that we find here in the Yukon, and actually from specimens that we received, from fossil specimens we got in the Yukon, specifically camels and bison. Okay, so just to go to the last ice age, this is the Earth as we know it today, focusing on the northwestern hemisphere. But during the last ice age, around 20,000 years ago, it looked a lot different. So we had massive glaciers covering a lot of the northern hemisphere, specifically the vast majority of Canada, albeit a bit of the Yukon not glaciated, and most of the rest of Europe, Northern Europe and Siberia. And as a result of so much water being locked up in these continental glaciers, sea levels are around 130 meters lower than they are today. So the keen-eyed around you might have noticed that, like, Britain is connected to France, Florida is a lot larger than normal, and of course, Beringia exists. So this paleo land bridge that existed between eastern Siberia and northwestern North America. So just to zoom in on the North American component of that, so this is height of the last ice age, 20,000 years ago. We have two major ice sheets, the Cordilleran and the Laurentide, and these two join together on the eastern flanks of the Rocky Mountains. Okay, so this is what North America looked like 20,000 years ago, and specifically in the Yukon, what we find now, kind of the remnants of that are during the last ice age, these are the kind of animals that you got in the Yukon. You had muskoxen, bison, horses, and mammoth. That was like the herbivore component. But you also had other predators like giant short-faced bears and scimitar cats. So this was during the last ice age, but prior to that, during the last interglaciation, which is the period in time, the warm period in time before the ice age, we also had other really interesting animals around, such as camelops, which is the camel that I'll be talking about, and also some other really interesting animals, such as the giant beaver, or Castoroides, which was actually around 2.5 meters long, so really, really large beaver compared to what you find today. All right, so the reason we're interested in the Yukon is one of the reasons it preserves fossils so well is because of this, because of permafrost. The permafrozen ground that you find throughout the Yukon, although not in this area, but as you go further north. And the reason we like it is because it preserves bones really, really well. So here we've got a mammoth tusk, part of a bison skull, and here's Grant with a horse skull, and you can see there's excellent morphological or anatomical preservation here. You can see it in the horn here, and you can see in the horse skull. But not only do you have excellent anatomical preservation in these remains, you also get excellent DNA preservation, which is why we like these specimens so much. But first, let's just step back and ask, really, what is ancient DNA? What is this all about? So let's just go back to just some really basic. So what ancient DNA is not is this, right? You cannot get DNA from amber-preserved insects, or specifically mosquitoes, and so we can't clone dinosaurs in this way. So no, this is not going to happen. So, and dinosaurs lived, up until around 66 million years ago. So what is the oldest ancient DNA? Reasonably, what have we got to? And the oldest DNA that we've actually found, and we managed to get a genome of this horse. This is a 700,000-year-old horse, which was a specimen that was actually found up in the Klondike out of Dawson. And actually, just this afternoon, I was with Grant and saw the specimen for myself, which was really, really cool. It resides in the Yukon government building, which is just literally a few blocks over on the other side of the airport downtown. And so we've got a 700,000-year-old genome, nothing older than that. So we don't seem to be able to get DNA that's older than 1 million years. And the reason we can get DNA preserved that's so old is because of some conditions that are really, really good for preserving the DNA, such as the permafrost, which we get here in the Yukon. And the reason it's really good for preserving DNA is it's cold and it's dark, so you don't have any ultraviolet radiation damage, and it's also very dry because the water is frozen. And really importantly, the environment is very, very stable, and that's really key to good DNA preservation. And that's another reason why when you hear about ancient DNA in the news and stuff, a lot of interesting results outside of permafrost come from cave systems, which are relatively cool. They're obviously not frozen, but they have very, very stable environments. Okay, so just going back to some really basic biology, this is what DNA looks like in a cell. You have these base pairs, which are A's and T's, C's and G's, and these are all wrapped up into these various around various histone proteins, into nucleosomes, and then compacted up into chromosomes. And the reason I'm showing you this is because in living cells, DNA molecules are tens of millions of base pairs long. They're really, really, really long. But DNA can get damaged in various ways. You can have chemical attacks, you can have bases changing, you can have base pairs being inserted, deleted, and so on and so forth. And out of all of these changes that can happen to living DNA, these are the 5 main things that happen to DNA in organisms that have died. So this is how DNA breaks down. And I'm going to talk about 2 of these, starting with double-stranded breaks. So here we have just like a very simplified DNA molecule. And if you have 2 single-stranded breaks, you obviously get some shorter DNA molecules. Now in life, this happens readily in living cells, but we have certain enzymes which actually basically glue together these DNA molecules that are broken as a form of DNA repair. But of course, if you're dead, you don't have those enzymes, and your molecules break down, and this continues, and what you end up with are lots and lots of short DNA molecules. So we start off with chromosomes that are like tens of millions of base pairs long, and we actually break down to really, really, really short fragments. So this is actually like what the DNA fragment lengths look like in the Yukon camelops that we looked at. Really, really short, so averaging only around 40 base pairs. So gone down from 20, 30 million down to only 30, 40 base pairs. And this is part of the reason why it's really challenging to work with ancient DNA. The second form of damage that I want to talk about are these abnormal bases, which make cytosines, or C's, that pair with G's look like these U's. So just very quick go over the biochemistry of this. If we start off with a cytosine molecule, which looks just like this, and you add water to it, so this is like a hydrolysis reaction, it loses an ammonia and it becomes this base called uracil. Now uracil is interesting because it doesn't ever occur in DNA naturally. But instead, when DNA is being replicated by various enzymes, as your cell divides, the DNA has to be replicated to split off. The replicator actually misreads the uracil and sees it as a thymine, it's a T. And so what this means is when you're sequencing this, what you see is this C to T change. And so this is a form of error, and this is another problem that occurs when we're looking at ancient DNA. And these errors tend to accumulate at the ends of DNA molecules, so I've basically highlighted these kind of like C to T differences that you see here with these little red dots here. So this is what ancient DNA looks like. Of course, when we're extracting DNA from a bone, if this is what your ancient DNA looks like, there isn't much of it, it's really short, it's really broken up, and it's been chemically modified so the bases look different. But your modern DNA molecules, so there's DNA everywhere. Modern DNA pollutes everywhere you go except for in the most extremely sterile conditions. So this can cause problems when you're trying to isolate your authentic, real ancient DNA versus contaminating modern DNA molecules. So for example, if these were ancient DNA molecules from like a bison or something like that, if you had contaminating modern DNA molecules from something closely related like a cow, that can cause real problems. So to avoid these problems, we work in like CSI forensic style conditions, such as this. So these are from various labs across the world. And this is basically how we have to dress up when we want to extract the DNA and really make sure that we keep the amount of modern DNA contamination to a minimum. So how do we actually extract, sequence, and reassemble ancient DNA? So we can use various tissue types, not just bones, for getting ancient DNA. You can use also teeth, even hair and skin tissues. But what's really exciting is, and this has really been developed in the last 10 years or so, is getting ancient DNA from actual sediments. So this is from dirt. In dirt, you get DNA molecules that have broken down from things that have died, things that have like shed their cells, like skin sheds off and things. And you can get DNA in the sediment, and you can actually extract DNA directly from the sediment. This is really interesting because the DNA composition of that reflects the entire community, whereas DNA from the bone is only obviously of the one animal that bone came from. In here, you're getting DNA from the entire community, so bacteria, the plants, the animals, everything that lived there. So this is really good for like potentially reconstructing ancient communities based on the DNA that's present. So we have DNA in the cases of the hard tissues like bone and teeth. We powder them down, and then we digest these into a buffer or a liquid, and we use various chemicals to do this, and this is basically to free up the DNA. But in this mixture, you've still got like proteins and lipids, which are fats and things, just various leftovers. So then we have to go through some filtration steps, and then what we end up is with a little DNA extract. So these are my fingers just for comparison. It's just this tiny little bit of liquid, and that has your purified ancient DNA inside of it. So how do we go from there? Well, what we next do is we modify our DNA molecules, and we add these artificial DNA sequences to the ends of the ancient DNA molecules. And the reason we do that is so that these DNA molecules can be read by the sequencing platform that we use, so the DNA sequencer. So these things called DNA libraries put on this sequencer, and the great thing about this is that these sequencers can sequence hundreds of millions of DNA molecules at the same time. So the output you get from the sequencer, and these short kind of like sequences of letters, these DNA molecules in the information, but you get millions or billions of these molecules. The great thing about having all that information is, so what do you do with all of this? So what you need to do is align it to what we call a reference genome or a reference DNA sequence. And this is just a known DNA sequence, and you basically map it. So it's like you have a jigsaw puzzle that's complete, and then you find one piece of the jigsaw puzzle, and you want to try and match it onto where it goes. So here's your reference DNA sequence. If we take these 4 DNA sequences that we know, we can actually put them along here. You notice how this all matches up along here. But there are 3 differences that you see here. There are 2 T's here, where at this position, where there was originally a G. So this looks like the individual where these DNA molecules came from actually differed evolutionarily from this reference sequence. So this is like actual DNA changes, so differences between the 2 individuals. And you can see this 3rd change I've highlighted in red here is actually one of these C to T transitions. So one of these, it looks like it's probably DNA damage. And another bit of evidence that it might be DNA damage is the fact that it's occurring at the ends of the molecules. So that's where that red dot was. Okay, so take this up. What we can do is we can collapse all of these into a new reference sequence. So this is basically identical. You've got some dashes here, which is where we don't have any information, so we don't know what was there. And the only difference between this reference sequence here and the reconstructed ancient DNA sequence is this 1 T change, which we see here. And this is great because we can then reassemble anything from just short sequences up to entire genomes using these methods. And then what we can do is we can compare these DNA sequences to what we find, to modern DNA sequences or other ancient DNA sequences we have. So what sort of analyses can we do with that? We can reconstruct family trees or phylogenies. So we can take the DNA sequence, look at how different it is compared to other DNA sequences, and then use that to infer relationships, phylogenetic relationships between individuals. So for example, this is a tree of humans. So here we have Asian and European and Africans. These represent all known modern human diversity. And what we have here are Neanderthals, these strange hominins that were only discovered a few years ago from an entire paleogenome, the Denisovans, and these really, really kind of like, we don't fully understand what they are. They're probably more closely related to Neanderthals, but we don't know for sure, these hominins from Cimodolosuesos. And as you can see, and all of these are ancient DNA sequences, and you can see from this we can say that Neanderthals, Denisovans, and these weird hominins fall outside of known modern human diversity. So that's something you can do with modern DNA. Another thing you can do is we are really interested in how migrations happen. So again, this is just like a human example because humans are a really excellent model for looking at migration history. We originated in Africa. And, some humans dispersed out of Africa. Some went to Europe. Some went down to Asia and Australasia. And then others went up into Siberia, across the Bering Land Bridge, and colonized the Americas. You can infer this pattern from looking at just modern DNA, but to really understand it, you need to have ancient DNA. And the reason for that is because not only do you have that sequence of that individual who lived at that time, but you can also date that time, so you can do radiocarbon dating. And that's really good for refining these ages of these dispersals. So if you have an individual from this time, they can tell you a lot about what's going on. So I'm going to apply this first kind of like approach of doing reconstructing a phylogenetic tree that we did recently, Grant and I and Beth and a few others, on looking at camelops and trying to understand what it is. So just a little bit about camelops. This is one of two groups of Yukon camels that you get, the other being Paracamelus, which became extinct nearer 2 million years ago. Camelops seems to only have existed in the Yukon probably for a brief time during the last interglacial around, probably around 180 to 100,000 years ago. So this was prior to the last ice age, but you still get its fossils preserved in the permafrost. So just a bit about camels and why we're interested in them. So interestingly, camels originated in North America around 40 to 45 million years ago, which is kind of, a bit weird because you don't see them here at all, which is funny because camelops actually lived up until 13,000 years ago and was amongst one of the last North American camels. So there were camels living in North America from around 40 to 45 million years ago all the way up to only 13,000 years ago. So throughout nearly all of their history, you had camels in North America. Then you've got the two groups of living camels. You have the New World camels, the ones in South America, llamas, alpacas, guanacos and their kin. And you have the Old World camels, the dromedary and the Bactrian camels. And the reason we were interested in getting, and the other interesting thing about camelops is although it was found in the Yukon around 100,000 years ago, it was the place where it's found in 13,000 years ago was south of the ice around Rancho La Brea and other sites around in the lower 48 United States. And there was evidence that it was among the species that were hunted. Not necessarily hunted to extinction, but they were definitely hunted, and that probably contributed to their decline. So we were interested in getting DNA out of camelops, and we had 3 specimens that Grant very kindly sent us from Hunker Creek, which is one of these gold mining localities in the Klondike. These are actually the 3 bones. Here's Grant holding one of these bones. They're all foot bones. And what we really wanted to explore with these DNA from these things is what is camelops. So we have these bones, so we should have some idea of what camelops is. How is it related to Old World camels, the Bactrian and dromedary camels, the 1- and 2-humped camels? Or how is it related to the South American camels, the llamas and alpacas, etc.? So here is a family tree of the South American camels here. So it's just represented by a llama, and the Old World camels. They thought to have diverged around 17 million years ago, so a really, really long time ago. And remember that camels originated would be way out here. And this is what the fossil record of camels looks like. So in red, I've highlighted all these kind of like extinct camel species. And you can see it's a bit of a mess. We don't have really very well resolved. We don't understand, when these things diverge from one another. And a lot of what we think we know, it turns out we really don't understand it that well. So just focusing on camelops, this is what we infer from anatomy, so from looking at just the bones. It seems that camelops is actually more closely related to South American camels rather than the Old World camels. But when we extracted DNA, when we reconstructed those ancient paleogenomes, aligned them to modern genomes, we actually found that camelops was more closely related to Old World camels. So completely the opposite kind of story that we were getting from anatomy. And so that basically turned the tree from looking like this, camelops closer to the South American camels, to up here like this. So this is what we understand of the relationships of camelops to modern camels now. And that's really interesting because, as I said, with that tree with all of the other fossils in there, now we don't really know where they sit at all. And so this will be really interesting for paleontologists to really try and understand how these extinct camel species are related to one another. And what we also want to do in future potentially is to look at some camelops samples from south of the ice and really understand more about the diversity of this taxon. Okay, so that's camelops. Now I'm going to move on to bison. And this is a paper that we very, very recently got published where we looked at the occurrences of bison in the ice-free corridor, which is this kind of like where those Laurentide and Cordilleran ice sheets were meeting, and tried to work out when the ice-free corridor opened. So a little bit, just skipping back to North America. This is what North America looked like around 23,000 years ago. So this is when this ice-free corridor closed. Prior to this time, the ice sheet, this is just prior to the last glacial maximum, this corridor was open, but at 23,000 years ago it closed, and this completely separated Alaska and Yukon from the lower 48 United States. Right here. So during this time, when Alaska and the Yukon were separated out from the lower 48, this is when humans first arrived in Alaska and the Yukon, probably sometime around 20 to 15,000 years ago. So this was prior to any humans occurring down in the lower 48. Then, around 15 to 14,000 years ago, humans came down from Alaska and Yukon and colonized the lower 48 United States and further south, and they spread really rapidly. And we know this from archaeological sites that we find in the lower 48. But the question is, how did they get down there? Did they get down there through this ice-free corridor that occurred between the Cordilleran ice sheets and the Laurentide ice sheets? Or did they take an alternative route? Maybe following the coast down, the Pacific coastal route down the side of British Columbia. And this is kind of like an unknown. No one really knows, and the reason that it's an unknown is because we didn't really understand when the ice-free corridor first really opened up. So if it opened up around 15 to 14,000 years ago, then that suggests that humans probably did use this route because that would match up with the timing of the archaeological sites we find. But if it seemed that it opened up less than 14,000 years ago, then that would mean that the ice-free corridor could not have been used for the first humans colonizing North America. So to answer this question of when the corridor opened up, to try and look at these ancient human migrations, we looked at fossils of bison from within the corridor. So bison are really interesting because during this time, the last glacial maximum 23,000 years ago, when the ice sheets had come together and there was no corridor, you had bison living up in Alaska and the Yukon and down in the lower 48. And what makes them an excellent group of animals to address this question is that they are genetically distinct populations. So they were isolated for long enough that you can tell if a bison is from the north, from Alaska and Yukon, or from the south, from the lower 48 United States. So what we did is we got a load of bison fossils from within the corridor region, spanning all the way from Inuvik down to Edmonton and Calgary down in Alberta. And we wanted to know, when the corridor opened, and we did this with radiocarbon dating and ancient DNA. So if you just did radiocarbon dating, so if you radiocarbon date these individuals, you find that the earliest individual is around 13,400 years old and occurs here in a site in Edmonton. However, just relying on radiocarbon data doesn't tell you where this bison came from. And that's important because did it come down from the north or did it come up from the south? It's simpler to say it came up from the south, but if the corridor was open, it could potentially have come down from both sides. Turns out that this individual that was 13,400 years old had actually migrated up from the south based on the genetics, which means that potentially this northern part of the corridor could still be blocked. Remember, for humans to come all the way through this corridor, the whole thing needs to be open. So you can have, if this area is open, then bison can come up, but you need bison to also come down. And to really identify when the corridor opens, what you really need to see is what the earliest time at which you find northern and southern bison in the same area. And that place is actually also Edmonton. And this happens by around 13,000 years ago. So this is our key bit of evidence of when the corridor seems to have first opened up. This is the earliest evidence. And of course, 13,000 years ago is too late for humans to have used this ice-free corridor region as the main mode of migration from the Yukon and Alaska down to the lower 48. So this may mean that this migration happened more along this Pacific coastal route, but it more or less rules out the ice-free corridor being the way that humans first colonized the areas of North America and South America south of the main ice sheets. Okay, so that's a bit of a bummer, but what can happen afterwards is you've got humans up in Alaska and the Yukon and down in the lower 48. When the ice-free corridor does open up, you can have migration actually in both directions, because if you've got humans up here, and then suddenly this potential route for people moving around opens up, they can go in both directions. And what's really interesting is you seem to get these kind of like these back migrations where humans may have migrated from the lower 48 back up to Alaska and the Yukon. And the reason we infer this is because around 13,000 years ago, when you have this Clovis culture and these really interesting fluted points, these arrowheads, just a little later than that, around 500 years later, you also see these arrowheads up in the north, so up in Alaska and the Yukon. So this is potential evidence for there being this backward migration from the south up into the north. Okay, so how are we doing for time? Okay. So, got loads of time, so I'm going to talk to you actually about a third project that we were involved in. This is the St. Paul Mammoth Project. So, a little bit about mammoths. You know, they lived in Beringia around here. So, during the last ice age, mammoths lived basically everywhere in the northern hemisphere. You got them from Europe, through Siberia, in Beringia, and also south of the ice. But as their populations were declining towards the end of the last ice age, prior to their extinction, the last places you find them is in Beringia, specifically on islands. So here is a map of kind of like modern day Beringia, so western Siberia, East Beringia, Alaska. And this red dotted line shows the outline of where the land would have been during the last ice age. And the very last mammoths who have ever lived here on Wrangel Island off Siberia until around 4,000 years ago. So this is really relatively recent, considering the rest of mammoths became extinct around 12 to 13,000 years ago. And this is actually at the same kind of time that the first pyramids were being built in Egypt. So that's a really interesting thought, that the pyramids were being built at the time when the last mammoths were still alive. So those are very last mammoths. But there was also another really late surviving mammoth population which survived on St. Paul Island here in Alaska. It's a really, really tiny island. It's only around 115 square kilometers, really, really small. And they survived there until, well, prior to our study, around 6,500 years ago. So still like around 5,000 years later than mammoths became extinct on the mainland of Siberia and North America. So a little bit about St. Paul Island. Here it is. This is actually the airport strip here, just to give you a sense of scale. It really is absolutely tiny. And the reason we were interested in St. Paul Island, okay, it's the second latest surviving mammoth, is to do with human hunting. So on Wrangell, 4,000 years ago, when the mammoth became extinct, approximately coincides with the first archaeological evidence of humans arriving. So they may well have had a role to play in the extinction of that island's population of mammoths. However, on St. Paul Island, there's a complete absence of archaeological evidence. And actually, the first evidence of humans ever arriving on St. Paul Island is during the mid-18th century, when Russian fur traders first arrived. So we had this population of mammoths that were going extinct maybe around 6,500 years ago in the complete absence of humans. So this must have been a completely natural, climate, environmental-driven extinction, which is really, really cool. And this is why we wanted to explore when and how the mammoths became extinct. So how do we know when the mammoths became extinct on St. Paul Island? I said to you 6,500 years ago, so how do we know that? And like how we know the timing of a lot of extinctions, we only know them from radiocarbon dating of the last known individual. Which is fine if you've got thousands and thousands of fossils and you dated a lot of them, and you can really, really have high confidence as to when the extinction occurred. Problem with the St. Paul Island mammoths is it's a really small island and there are very, very few fossils. So you only get like a handful of radiocarbon dates. So you can't have very much confidence in if that last radiocarbon date you got was, near when the last mammoth became extinct, or if it's just a sampling artifact, if you just haven't got enough samples to really be able to say anything. So this is where we took this kind of like the sedimentary ancient DNA approach. So as I introduced earlier, you can get sedimentary DNA preserved in mud and dust, and that's been permafrozen, but also in lake sediments too. So on St. Paul Island, we actually used a lake sedimentary DNA approach. So this is just the basically buildup of the silt in lakes. And the reason we wanted to do this approach is because mammoths, like all elephants, drink a lot of water. So they have to go to water sources regularly to drink a lot. And when they go to water sources, they go in, so they're shedding skin, they're shedding hair and other bodily fluids into the water. And so you get DNA building up in the lake, and that DNA settles down and it settles into the lake sediment. So that is how the DNA gets into the lake sediment. And what was really great is in the middle of St. Paul, there's actually a lake, a crater lake, which has been around since around the late Pleistocene, since around 15,000 years ago, and has a continuous sequence of this sedimentary column. So you have basically DNA of all of the animals that were living through time from 15,000 years ago to the present. And obviously that spans the 6,500 year window where we think the mammoths went extinct. And this is what the lake looks like. As I said, it's a crater lake. It's around 200 meters across, but at its deepest point, it's only 1.5 meters deep. So it's a really, really shallow lake. It's like a saucer. And from this, so that's the water depth, but the depth of the sediment underneath, so the lake sediment, is actually around 13 meters deep. So you have 1.5 meters of water and then 13 meters of the sediment underneath. And what we did is we drilled through that sedimentary core. Obviously doing that at this time of year, during the summer, is problematic. You'd have to go out on a boat and it'd be a real pain coring down 13 meters. So the obvious thing to do is to go out there in the winter. So this is actually on Lake Hill Lake. I didn't personally do this. This was some of our collaborators. They went out onto the lake, it's covered in ice, and they cut through the ice and they rammed cores down into the sedimentary column and collected this 13 meters of sediment. And then once they'd taken that out, we took it back to the lab, and we basically took these samples through the core. So you got 15,000 years of history spread through 13 meters of core. If you start sampling this core at very, very fine intervals, so around every 4 centimeters here, this is looking at the time. The time you're looking at here is on the level of several decades to maybe a century. So you're looking at really, really fine time sampling throughout this. So that's great. You can you've got the sedimentary column. You can take samples that you know how old they are, but how do you identify mammoth DNA from the sediment? You know, you've extracted DNA from the sediment. As I said, there's going to be plants, bacteria, other animals in there. But how do you identify mammoth? Because at the end of the day, DNA is the same amongst all life. It's all got A, T, C's, and G's. So how do you do this? And what we had to do is we had to develop a number of novel computer-based methods to deal with this. So here are those sequences that I showed you before. And what we did is we aligned them to 2 different reference genomes, so these reference sequences, the African elephant and the woolly mammoth. So that's the first way of doing it, is to see, you know, if your sequence aligns to these genomes. And that's good evidence that it might well be from mammoth, or if it doesn't, then it probably isn't. And just to check that this was kind of like, you know, running as we would expect, we also aligned our sequences to the 2-toed sloth. The reason we did this is because sloths didn't exist in Beringia at this time. Giant ground sloths did exist in Beringia, but they became extinct during the last interglaciation around 100,000 years ago. So we shouldn't expect there to be any sloth DNA in here. And this is important to do because it kind of provides you with a control, so to understand, like, you know, if I just gave this system any random genome, what would I get? And so we took all of the kind of like DNA sequences that align to these 3 genomes, and we did some further filtering, so we got rid of anything that also matched horse and cow, mouse and human. And the reason we did this is because these are common kind of like laboratory contaminants. They're all mammals, so you might identify the DNA wrongly. And we also looked at this database, which is of many DNA sequences that we know, including bacteria, and sometimes you can accidentally align like bacterial DNA to these reference genomes. So we wanted to remove that as well. And then you end up with just a small number of DNA sequences which we have high confidence are mammoth, and then you get very, very few which kind of still get through that look like sloth. They're not actually sloth. What this is telling you basically your rate of error of how many things just get through randomly. And one of the other ways we can look at this is we can look at the DNA length and the amount of DNA damage. So this is the plot that I showed you before from the camels. You can see you usually get this peak around 30, 40 base pairs, and that's a good indication that your DNA is ancient, usually less than 100 base pairs. And what this is showing here is that signal of DNA damage where your C looks like a T, and this is basically this red line here is showing you that the amount of damage that you get increases as you get closer to the ends of the DNA molecules. So what you want to be looking for if your DNA is ancient and authentic is you want to look for a distribution that looks like this, fragment lengths, and you want to see a red line that goes down like this. So what do they look like when we align to African elephant, mammoth, and 2-toed sloth? They look like this. So here's all the African elephant, mammoth, 2-toed sloth. You get these nice distributions for the woolly mammoth and the African elephant, not so much with the 2-toed sloth. And likewise with the DNA damage signals, you get this increase in the red, which means you're getting more of these cytosine to thymine mistakes occurring at the ends of molecules, and you see this in the African elephant and in the mammoth, but then it's a complete mess in the 2-toed sloth. So this is really great because this is showing you that we're seeing these authentic, these real kind of like ancient DNA signatures from the things that we think are mammoth, but not from the sloth. So that's really, really great. So what did we actually find from these samples? So here we have the core depth. So as you increase the core depth, you're going back through time. So 0 is your surface, and then you head down the core, and that's basically tracking back down in time. And what we see is that the amount of mammoth DNA, shown here with the red and the blue, actually drops out completely at around 500 centimeters. And this actually corresponds to around 5,600 years ago. So you remember I said to you, based on the last radiocarbon dates, it was around 6,500 years ago for the mammoth disappearing. What this sedimentary ancient DNA is suggesting to us is that mammoths actually became extinct on St. Paul nearly a millennium later than we originally thought, so around 5,600 years ago. And what was really cool about this approach is when we looked at this core, we weren't just looking at DNA, we were also looking at other things like pollen and spores. And the great thing about spores, fungal spores, is they occur in animal dung. And we see also a real big drop-off in animal dung spores at exactly the same time interval. So this is a kind of like an independent line of evidence that mammoths were disappearing from St. Paul at this time. Okay, so just to summarize what we've learned in this talk, that camelops is actually more closely related to the Bactrian camel than to the South American camels. We learned from looking at bison fossils that the ice-free corridor opened around 13,000 years ago rather than any earlier, around 14 to 15,000 years ago, which means that it probably was not used by the first humans colonizing from Alaska down into the lower 48, but could well have been used for later migrations going back up. And we've learned that the woolly mammoths of St. Paul became extinct a lot later than we originally thought. And just to finish up, I just want to thank my various members of my lab, especially Beth, James, Andre, and Josh, for all their help in assisting with extracting and DNA in various discussions, and also all of my co-authors and collaborators, especially Grant, who's here, who provided a lot of the samples that we used for most of these studies, and also the gold miners of the Klondike, who basically provided us all the samples. If it wasn't for these guys, we wouldn't have known anything about camelops because we got all of those specimens from the Klondike gold miners of Dawson. And with that, thank you very much.
Question. What the core samples that you have in those trays, what's the texture of it? It's like shale or hard sand, or is it?
It's all lake sediment, so it's basically mud. So depending on which depth you were throughout those 13 meters of the core, some of it is very silty, and then others gets very kind of sandy. And then in other areas, because there's been some volcanism through this time, you also get tephra layers, these ash layers sometimes, and then there's also some basaltic, kind of like much darker sandy layers as well. So it varied as you went through the core, but most of the first 10,000 years of the core is mainly this kind of like silty, organic rich mud.
And do you keep it frozen?
So interestingly, that lake sediment, it's not actually frozen at all. That it's more kept at like refrigerator temperature. So that lake, the core was never frozen, so the permafrost doesn't ever reach that far on St. Paul Island, that depth. But yeah, so it doesn't stay frozen, so we didn't refreeze them at all. But it's cold enough to preserve the DNA.
So were you able to tell why they went extinct?
Yes, so that's part of that's an interesting part of the story that was told by some of these other kind of like proxies and lines of evidence. So I talked about the DNA and the fungal spores, but we also looked at pollen and various isotopes and various plankton and other things that were preserved in these cores, and you can tell a lot about the paleo environment that was going on. And what seems to have happened at around this time, around 5,600 years ago, is that the lake was becoming much saltier, and it was becoming much brackish and drying out. So we think that actually the mammoths just couldn't drink the water anymore.
So did that lake sediment show how vegetation might have changed?
So from the pollen records, yes, there have been changes in this in the vegetation. What was interesting is through that period where the mammoth disappeared, the vegetation actually stays really constant. So that was a way we excluded like really major kind of like environmental changes causing this extinction. We've actually looked at DNA from some of the plants. So using this approach, obviously we just looked at mammoth in this study, but you can also look at plants and any other kind of like organism you want. And the results from that were pretty consistent with what we found from the pollen, where throughout most of the core, the vegetation was consistent, and you just got minor changes here and there.
Are there freshwater sources on Wrangell Island that could be used to compare that whole brackish water idea? Think you guys would know?
That is a good question I do not know the answer to.
The extinction on Wrangell, was that when humans got there to hunt, or was that also an environmental extinction?
That is a very good question, and we don't really know. So it's really hard to disentangle whether extinctions are from almost certainly due to a combination of human hunting and environmental changes where you have both of those factors occurring. What made St. Paul really interesting is you didn't have that confounding factor of human influence causing the extinction.
As the permafrost is melting, is that degrading the DNA samples that you could possibly?
So, yes, when the permafrost melts and samples kind of like fall out of the permafrost and they're no longer frozen, the DNA just will start to degrade at a much faster rate. And we've seen this where samples have been taken out of the permafrost and stored in different museums, and sometimes they're stored in, really well stored, and you can get DNA out of them like 50 years, 100 years later after they've been put in the museum. Then they're stored in other museums where the environmental controls are not so good, and you can't get DNA out of them. So it's amazing once you pull them out of the permafrost, it's pretty important how you store them. The flip side is, as the permafrost melts, it's actually exposing more and more samples for us to get. So from that perspective, in a way, it's kind of good for us in that we can get some more samples, but long-term wise, no, it's not that great.
So in a lake core like that, how can you account for migration or upward or downward movement of particles within that sedimentary column? So, yeah.
So that can absolutely happen, but we were pretty confident that we weren't getting too much migration. Are you talking about DNA specifically? Yeah. Or? Right. The reason we don't think that is because we got such that sharp drop-off. So so where we see mammoth to where we don't see mammoth, that is a 2-centimeter window. So it goes like that, and we sampled 2 different core drives that overlap that region, and at exactly the same point, the mammoth DNA dropped out. So that seems to suggest that you weren't getting migration of DNA, which just happens when you're in a non-frozen sediment. Your DNA is free to move potentially, and it can move up and down the core. Obviously, that's moving into parts of the core that are of different ages, and that can cause you problems. But we don't see that so much in the lake hill core. Not at all.
Sorry to speak from the eaves, but that ice core is really fascinating in the sense that you might be able to say what the mammoths lived on during the winter months, and whether that sort of food stock disappeared at their extinction, or whether it
As I said, the vegetation on St. Paul doesn't seem to have changed too much. What does seem interesting is from the bones that have been radiocarbon dated, we've also done isotopic work and looked at nitrogen isotopes. And that seems to indicate that there was some, not necessarily dietary shifts, but they were getting a bit more water stressed near the extinction event.
Water stress as in no water.
Yes, yeah, or less water to drink.
Does that mean say it's freezing up during the winter? Or?
No, it's part of this. So as I was saying about from the looking at the paleo-environmental proxies, they were suggesting that the lake, which was their main water source on the island, was gradually becoming saltier and more brackish. So they were just having less and less to drink, and then it got to a tipping point where mammoths just couldn't survive there. And that seems to coincide very nicely with the extinction event. Of course, inferring kind of like exact causes of extinctions is very, very hard. But the beauty of this study was that we had this really fine time scale, so we could really well correlate these events that were occurring.
It's always been a fascination to me what mammoths live off in the winter. They're massive animals, and if you see them in Africa, they eat like shitloads every day, destroy anything that's edible.
How, considering this brackish, the concept that the water's picking up salt, probably windblown or something from the ocean or whatever, how salty is that lake now?
It's not that salty. It's pretty fresh based on the inference of how salty it was based on the plankton. So the cladocerans, these plankton along with diatoms that you get, and as I said, at the extinction window, we see this increase in the more salt tolerant diatoms and cladocerans. As for what you get there now, it seemed pretty fresh water, but we didn't really look at exactly what was going on today as part of the study. So I don't know exactly how brackish it is. It's not that brackish, but...
So how did the salt, because at that point in time, excuse me, the water levels, or the sea level should have been 6,000 years, I suspect, is about what it is now.
Yes, the islands, so the island size, I didn't actually discuss the island size, but the island attained its kind of like close to its current size around 2 or 3 millennia before they became extinct. So it was unlikely to be like seawater intrusion, but why it became salty, I don't really know.
Well, a lot of marine sediments are probably being deposited as loess into that basin as well. So I'm just making it up.
Yeah.
Yeah, that would be something to look at, whether what the salt was, whether it's salt of marine origin or like our salt flats out here, which are the salt is trapped in sediments, but it's not sodium chloride.
That's interesting. We never looked at the kind of like ionic content to try and work out what if you can do that. I'm not an expert on this, but I don't know if can you do that.
You should be able to tell whether it's whether
Okay.
it's apholastic or whatever, but non-marine origin.
Ah, interesting.
And so, which it would become saltier because you're getting more evaporation. So
Right.
that's what's happened out here. Whereas if it was, there could have been some big event where you're getting big storms or something that are putting more salt onto the land. And so there might be, that would be another story.
And also your different salts will precipitate out at different concentrations. So for instance, the open prairies, they can actually, you can tell by the different layers, like potash, or those salts come out before sodium chloride. They actually get layers of these different salts, and that'll give you an idea of why those salts maybe where they are.
Okay, that's very interesting. Thank you for
Because you've already
that
got
feedback.
those.
Yeah, absolutely.
I know that there was the tendency, like on the Channel Islands off of Santa Barbara, for the mammoths to become pygmy. That's a trend in island ecologies. I don't know if there's any references that was that occurring during this phase, because there's some argument that some of the higher Arctic were experiencing the same kind of tendency.
Yeah, absolutely. So on Wrangel Island, that absolutely happened. So there was island dwarfism, where you get populations marooned on islands, and they just become smaller as they adapt to their habitat and to survive. Yeah, that absolutely happened on Wrangel. The mammoths on St. Paul were the best preserved specimen actually is from a pitfall cave, where you have just an opening, a lava tube opening, and a mammoth had fallen in, and we've got a fair amount of the skeleton from that. Unfortunately, it's I believe it's a sub-adult or a small female, so it's hard to tell how large they actually got. Again, there's not a great fossil record on St. Paul, so there are just a few isolated elements.
Genetically be able to be determined in comparison to Wrangel or other?
You can compare how the St. Paul Island mammoths are related to Wrangel Island mammoths, but you would use that from the bone and tooth data rather than the sedimentary DNA data. The problem with the sedimentary DNA data is you can't tell how many individuals contributed to that. So you assume it's one population because it's an island, but yeah, it could be multiple individuals. So you can't do any fine level population genetics in that respect. But using the bones, you can work out how the Wrangel and St. Paul Island mammoths were related to one another.
Totally unrelated.
Okay.
It's been driving me nuts ever since you put this slide up. What is a flat slug?
It's a banana slug, anything but a bear.
This guy knows. Yeah.
It's obviously related to your university.
Yeah, so this is the UCSC mascot. Like, absolutely, it's the banana slug. So originally it was a bear, but then the students said, no, we want the banana slug to be our mascot. So it's native to California, and it's found in the redwood forests where the UCSC campus is. And when it very occasionally rains, you get these banana slugs come out, and they're really bright yellow. Really interesting, cool things. Oh, really?
Oh, yeah.
Oh, I didn't know they could that far enough. Cool.
Sorry for that.
No. It's good.
What's the next project?
Oh, Grant.
What's the right answer?
The right answer is horses. So horses are the next big thing I'm working on with Grant.
That's it.
That is a story for another day.
Okay.
All right, cool. What was your question?
Core sampling like that along the coast?
Of like Alaska or? No. So.
Type of DNA we're finding on the BC coast and the Alaska coast.